Hello and welcome to the General Microbiology course. This course is part of the Agronomic Science program at the Department of Natural and Life Sciences and Earth Sciences at the University of Buira. It is presented by Dr. Linda Makhloufi and it belongs to the fundamental unit with 6 credits and its coefficient is 3. Microbiology is essential science that will help to better understand the invisible world and how it impacts agriculture, health, environment, and biotechnology. For the course outline, we have 3 chapters. First, the microbial world, then the characteristics of bacteria, and finally, the concepts of mycology and virology. By the end of this course, we will be able to achieve several important objectives. First, you will be able to understand basic microbiology terms. Then, we will focus on how microorganisms are classified so that you can recognize the major groups. After that, you will learn the difference between prokaryotic and eukaryotic cells. You will also look at the structure, nutrition, and growth of bacteria, which are center topics in microbiology. And finally, you will be introduced to the concepts of fungi and viruses to understand how they differ from bacteria and other organisms. The chapter 1 is about microbial world. Let's start by talking about the definition of microbiology. Microbiology means the study of small life. Now, what are the microorganisms? Microorganisms are tiny living things that we usually can't see with the naked eye. This chapter introduces the different branches of microbiology, such as bacteriology, virology, mycology, parasitology, and others. We also learn where microorganisms fit into the tree of life. They can be classified as either prokaryotes or eukaryotes. Prokaryotes have no true nucleus, while eukaryotes have a nucleus. Then we go over taxonomic classification. The system used to organize all living organisms. It starts from the kingdom level and goes down to the species. The second chapter is about the characteristic of bacteria. In this chapter, we focus on bacteria. We study how they grow in the lab and the techniques used to observe them with the naked eye and under the microscope. Then you will learn about bacteria shapes and arrangements such as cocci, bacilli, chain, and clusters. We'll explore the internal and external structures of the bacterial cell, such as cell wall, membrane, ribosomes, nucleoid, pili, flagella, and more. Some bacterial structures such as DNA and the cell wall are always present. Others like endospores, capsules, or flagella appear only in certain bacterial species. Bacterial growth is influenced by 5 main factors. We have water, temperature, pH, oxygen, and nutrient availability. Then we look at bacterial nutrition and how they get energy, carbon, and other elements. Let's move on to the bacterial reproduction and how cells grow. Bacteria grow by binary fission. They double their component and split into 2. To understand how bacteria grow over time, we use something called a growth curve. In a closed liquid culture, bacteria growth follows 4 phases. We have lag phase, we have log phase, we have stationary phase, we have death phase. This figure shows the 4 phases of bacteria growth. At first, when microorganisms are introduced into a fresh culture medium, they don't start growing immediately. This is the lag phase. During this time, cells are metabolically active but not dividing. They are busy repairing themselves, synthesizing ATP, enzymes, and ribosomes to adapt to the new environment. Since the medium is different from one they were used to, they need time to adjust before starting growth. The second step, known as the log phase, is when cells start dividing quickly in a logarithm pattern. Microorganisms grow at their maximum rate and conditions such as nutrient and temperature are optimal. This results in a population of identical cells dividing at regular intervals. This is the most stable and uniform phase, which is why it is often used in biochemical studies. However, as nutrients begin to decrease and waste products accumulate, the growth rate starts to slow down. At this point, the culture reaches a balance, leading to the third phase. During the stationary phase, the number of living cells stays constant. This is because the rate of cell division equals the rate of cell death. Our cells stop dividing but remain alive. The main reasons include nutrient depletion and we have oxygen limitation or accumulation of toxic waste. In the death phase, the number of living cells starts to go down because the environment is no longer good for survival. A cell is considered dead when it can't grow or reproduce anymore, even if it is still looks normal. This phase shows a permanent loss of the ability to grow. In the last chapters, we covered the basics of mycology and virology. Viruses are tiny, they are not cells and can only reproduce inside a host. Then we study molds, which are multicellular and form spores. Some molds are useful in biotechnology, while others can be harmful. If you have any questions or comments about the course, feel free to contact me. I'm always happy to support you in your learning journey. Thank you for your attention, and I hope you enjoy discovering the world of microbiology.